Today I'm going to tell you a little bit more about GREAT for preclinical animal studies, as Alexander already introduced you to. But first a little bit about me, although this is now, most of it is already said, but I'm an assistant professor at the meta research team at the Radboud University. We're part of the Dutch CAMARADES Center in the Netherlands. And in my background, I studied biomedical sciences. I did my PhD a very long time ago into the effects of nutrition in Alzheimer's disease. And after that, I started to work in the evidence-based medicine field, and I started to focus on methods for preclinical systematic reviews since 2010. Our research focus in our group is mainly on the experimental animal studies, but also the in vitro studies. And we're trying to expand more towards the human studies as well. We are mainly busy with developing methods for systematic reviews and meta-analysis. We have been developing guidance for meta-analysis. We started to work on risk of bias tools, develop search filters to identify animal studies, etc. We created protocols for pre-registration of systematic reviews and various reporting guidelines. A lot of what we do with our team is also training and coaching into the conduct and validity of primary research, so the actual animal studies itself, but also in the conduct and validity of systematic reviews. Now, if you are inspired and you want to know a little bit more, you can look up this website later on, and it tells you what we do, and it provides you some of the tools and other things we published over the years. Okay, so now actually I think it's time to start. And since you subscribe for this seminar from COReS, and the aim, of course, as Alexandra just mentioned to you, is to integrate preclinical systematic reviews in the research pipeline when translating scientific findings, I assume that you're already a bit familiar with systematic reviews, although I just noticed that it's just a few of you that already conducted a systematic review. But therefore, I will not say too much about definitions of systematic reviews and meta-analysis, but I will briefly describe the main steps before starting so that we all start at the same page. So when you conduct a systematic review, you officially, of course, start with phrasing the research question, and you write all the details regarding your methodology in a protocol for systematic reviews of animal studies. And subsequently, you can start to search for all available evidence. So generally, you do that in multiple databases, for example, PubMed, Web of Science, etc., using a very comprehensive search strategy. So trying actually to obtain all the published peer-reviewed literature regarding your topic. When you identified all the available literature, you need to start to screen which papers are actually relevant for your systematic review, and you use predefined eligibility criteria to do that. Now, after that, you extract characteristics, you extract or assess the quality of all the available studies, and then finally you'll end up with an overview of the situation. But if you have this overview of your field and you have maybe conducted a meta-analysis and you have a point estimate, we need to know how reliable your entire systematic review was. There are always subjective decisions. There are always steps where we're not really sure about. So at the end, when you also summarized your evidence, it's time to evaluate the actual results of your systematic review and assess what we call the certainty of the summarized evidence. And this will help you to improve the translation of the results of your systematic review, often a preclinical systematic review, to the actual clinical situation. So, but why should we make a thing about evaluating the results of a systematic review? Because most of us probably think that if we have this systematic review, we're done. Now, first of all, systematic reviews of preclinical research form the basis of clinical studies, and the interpretation of those results should therefore be done reliably and reducible. Second, the interpretation is not always as straightforward as men may think. So a variety of factors can be taken into account, and also a variety of factors should be taken into account. But which factors are important? Which one do we need to take into account? And how do we do these things structurally? Now, research showed that there is actually a large variety in how scientists interpret the results of their systematic review. And sometimes this leads to different conclusions from actually the same data, which may lead to confusion and errors in practice. So, for example, I borrowed here an example from the clinical field, but it highlights the importance of a structured way of interpreting the evidence. But I borrowed, as I mentioned, an example from the clinical field about use of beta blockers in hypertension. And in the clinical field, in the early 80s, a meta-analysis was published suggesting that beta blockers reduce mortality in hypertension. And this led at the time to routine use of these beta blockers in clinical practice. However, much later in time, larger RCTs showed that beta blockers were less effective than other hypertensives. For example, think about the ACE inhibitors, diuretics, etc. And they were just less effective in reducing stroke or cardiovascular mortality. So what went wrong in this initial systematic review? Now, actually multiple things went wrong. So for example, the early systematic reviews, they failed to consider important factors like subgroup variation. So later systematic reviews showed that the beta blockers may be more effective in some patient populations. So those with, so I see my Windows thing, wait 1 second. It's gone. So what was I saying? Yeah, I was talking about subgroup variation, and that the beta blockers were more effective in specific patient populations like those with heart failure. But the early systematic reviews treated all hypertension patients as one single group. Another thing that they didn't take into account was the presence of, for example, indirect evidence. So the early studies, so the early SRs, were often based on indirect comparisons, such as trials conducted in younger or healthier populations, rather than those with cardiovascular risks or other comorbidities. And that's, of course, very important to take into account and provides errors when you want to translate those results to the actual clinical practice. Another factor that was not considered structurally was bias and inconsistency. So, for example, the early meta-analysis didn't systematically assess the quality of the evidence or addressed inconsistency between the study results. And GREAT, the system where we're going to talk about, would have flagged these issues and likely downgrading the strength of the evidence later on for actually, in this case, inconsistency and risks of bias. So this example tells you that there are multiple factors that need to be taken into account, and we need to know which factors and how. And this is why there are multiple systems developed. So to make sure that the interpretation is nuanced and takes in all important factors in account, we need a structured, standardized, that's always a word which I struggle with to pronounce, but we need a transparent method to evaluate the certainty of the summarized evidence. Now, as mentioned, there are many systems to assess the certainty in the evidence, and a few are mentioned in this slide. Now, today we're focusing on the structured qualitative tools. In other words, those are more like domain-based tools. So we focus on these domain-based tools as they form also the basis for most preclinical certainty in the evidence assessments. Now you see various tools mentioned in this category. So this is because all these tools are tailor-made for various research fields. So they are all GREAT or GREAT-like approaches. So the initial one is the GREAT, which is developed for the clinical field. And I will tell you more about that in a second. The navigation guide is GREAT-based, and it was mainly for environmental health studies. OHAT is more for the toxicological field, includes animal studies, but also in vitro studies. And our circle framework is mainly developed for preclinical animal studies. But to explain the circle tool, which we developed a few years ago, I start with explaining actually what GREAT is and which factors or domains play a role. So GREAT stands for Grading of Recommendations, Assessment, Development, and Evaluation. So a lot of words. And GREAT helps us to evaluate how confident we can be in the results of research and their applicability to patient care. So that was the original thought. So here, for example, you can see a typical summary of the evidence table from GRADE, in which you see multiple things. So first of all, you see a list of the important outcomes. No, I should start here. You see the research question. So you see that in this table, it's about non-selective beta blockers compared with placebo for primary hypertension. Subsequently, you see all the various outcomes, and then they have desirable and undesirable outcomes. You see a measure for their typical burden, so the actual effect size. You see the number of patients that are assessed in the meta-analysis for all these specific outcomes. And you see here the quality of the evidence for each specific outcome separately. So what we are going to discuss is how did they reach these quality of the evidence ratings and which factors are playing a role. Now here you see depicted how the certainty in the evidence systems, the process actually goes. So per outcome of a systematic review, you will assess the role of various domains. And domains that lower the certainty of the evidence are downgrading factors, such as you could see here, risk of bias, unexplained inconsistency, indirectness, imprecision, and publication bias. And there are also some domains that increase the certainty of the evidence. We call that upgrading factors. So think about magnitude of effect, dose-response relationship, consistency, for example, consistency between species in case of experimental animal studies, and other ones. Each outcome starts with an initial confidence rating. So here at the left side of this slide. So RCTs start as a high initial confidence. So controlled animal trials would do the same. And observational studies with, in which there's a lot more confounding generally present, they start as at a lower level. Subsequently, the next step is to apply per outcome if you think there's a risk of bias, inconsistency, etc., and whether or not you want to downgrade the certainty in the evidence, but also whether there are factors that increase your confidence in your outcome on your diamond, for example. And this is actually what you do, and then you get an overall rating, a final confidence for each specific outcome, which could be high certainty, moderate certainty, low certainty, or very low. Now, I want to dive a little bit more into the domains that play a role in preclinical animal studies. So first of all, risk of bias. And because most of you are probably familiar with systematic reviews, you know that in a systematic review, we try to assess the actual risks of bias in the study. So for each study that is included in the systematic review, we assess whether there is a risk of selection bias, performance bias, detection bias, attrition bias, etc. And for all these studies, we have these rating, and the next step is to check if we have this overall idea about risk of bias, how much this interferes with the conclusion we were drawing from our systematic review. The next factor is about inconsistency. So how much heterogeneity do we have between the studies that are included in our systematic review? There may be different population groups, etc., that should not have been pooled. Could we explain the inconsistency or the heterogeneity between the studies? This is the second factor. And don't worry for now, I will go into much more detail all of these factors in a minute. The imprecision is one important factor. When we're talking about imprecision, we're talking about the actual precision of the diamond of your summary effect size. So how much trust do we rely on how precise the direction of effect is? Indirectness, which is about whether or not we trust that the study population studied in our systematic review is the same as the population we are trying to target in the actual clinical situation. Publication bias is a reason for downgrading, because if you miss important studies which show negative or neutral effects, the overall effect of your meta-analysis is often generally overestimated. And of course, in preclinical grading, we have upgrading factors. One of the most important ones is the consistency between species. So if you want to translate your results from the preclinical field to the clinical field, we generally have more certainty in our evidence if all the species that we identified or studied in our systematic review have the same direction of effect. That will increases our certainty that it will actually work in the same way or have the same direction of effects in the human population. Sometimes we also take dose response into account. So for example, if you have these studies with a low dose, other studies with a high dose, etc., and you all pooled all of these studies and there's a very clear dose response relationship available or present, then we also rely a little bit more on the actual effect that we are observing. Okay, so now let's start with the first factor in more detail. And maybe if I can't see you all, but if it's going too fast or you have questions in the meantime, don't put them in the chat right now because I can't see them, but you are allowed to speak up, for example, or if you want to put them in the chat, I would like maybe Alex or Frederic to notify me.
Yeah.
It would be very useful.
We'll monitor the chat.
Let's talk about this first downgrading factor, risks of bias. So when we're talking about bias, we're talking about in the context of systematic reviews, we're talking about systematic errors made generally, in general, unintentionally by the researchers. So systematic differences between the experimental and the control group, which are not due to random error. So we're talking about when there is bias, we often have less certainty in the actual evidence provided in the study. And when you have a low methodological quality, which causes bias in the study results, you often have an overestimation or an underestimation of the true intervention effect size. Here you see a paper, I think it was definitely from CAMARADES in UK, and I think from 2008. I'm sorry that I forgot to put the reference in, but what you could see here is that they clearly showed that if you have studies that actually randomize the animals into their groups or did not randomize the animals across groups, that the actual effect size is very different. So the effect of this specific outcome, which was in this case reduction in infarct volume, is smaller in studies that randomized the animals across the group than in studies that did not randomize. I'm not sure if you are all aware on how to read this, but what you could see in this gray horizontal bar in a way is that this is the summary effect. So it's the diamond of your meta-analysis. And these are subgroup analysis. So studies, all the studies that randomized the animals and all studies that did not randomize. And then you can see that the overall effect has a certain X effect size, and the randomized groups, which actually is the right way of conducting your experiments generally, showed smaller effect sizes. The same is for whether or not you blinded the outcome assessment. If you did blind the outcome assessment, often the effects are smaller than in studies that did not blind the outcome assessments. So what is important to realize is that the conclusions of your systematic review, they depend on the quality of the included studies, which is of course quite obvious, but just to make sure. So this is why we actually conduct a risk of bias analysis regarding the included studies in our systematic review. Now, in grading, when we want to interpret the evidence or the conclusions we have drawn, we want to check what it actually did with our certainty in the evidence. So in a way, we're looking at the actual evidence, all the evidence we found, and we want to know if this evidence may be biased. So we are now no longer talking about assessing the risk of bias of the individual studies, but we're now talking about assessing the risk of bias of the body of evidence. So, for example, if you have a research question, let's do it differently. Here you see, for example, a risk of bias analysis, and you see that most circles are green, indicating a low risk of bias. For one aspect, blinding of the data analysis, you see a lot of red. So most studies did not blind the outcome assessment, And this, of course, may be risky, but now when we are assessing how to interpret the certainty of the evidence, it depends first of all on the type of outcome you're assessing, but also on whether this is serious or not. So, for example, if the study investigated the effect of an intervention on, let's say, mortality, then I'm not so much worried, because mortality is quite clear, of course. However, if this systematic review was about an histological outcome, then all this red is much more to worry about, and then I may want to downgrade my evidence because it may have been subjective or overestimated. So what is important is that when you are assessing the domains for grading or interpreting the certainty of the evidence, you're no longer talking about individual studies, you're looking at the actual body of the evidence. So all the studies together, and you assess whether or not the risk of bias that you observed may influence the conclusions that you draw. And that is actually what you do. I hope that's clear. The next domain that you use when you want to interpret the evidence is inconsistency. And inconsistency is actually the same as heterogeneity. And when we're talking about heterogeneity, we're talking about excessive variation in the results of studies. So there will always be some variation, often due to the various sample sizes and thus random error. But this is about excessive variation between the studies. And when you're assessing whether or not there is inconsistency playing a role in your systematic review, you assess if there is still substantial heterogeneity after exploration of your a priori hypothesis that might explain heterogeneity. So I'm talking about subgroup analysis, for example. So if you have a subgroup analysis that may explain the heterogeneity, you do not want to downgrade. But if there's still, after meta-regression or subgroup analysis, a lot of remaining heterogeneity, we are less confident in the conclusions that we are drawing. So when you are looking at statistical heterogeneity, you need to take a few things into account. So first of all, you look at your forest plot, and then you check whether or not there is a large variation in the actual effect sizes. Another thing that you are doing is that you check whether or not there's a lot of overlap in confidence interval between all those individual studies. And of course, you can look at heterogeneity measures, the I-squared, for example, and a statistical test. So again, here also for this domain, and it's for all domains, think about the body of evidence and try to judge if the observed or the remaining heterogeneity may reduce our confidence in the results. So I understand that this sounds all difficult and challenging, and it took me also quite some time to really understand how this grading system works, but I try to explain it a little bit more in detail. So here, for example, you see an example in which I want to try to explain inconsistency. So you see here 4 studies, and they all are on the left side of the plot. So they all have the same direction of the effect. So in a way, if we consider this variability substantial or not, depends on the review question. So for example, is our question about efficacy or harm? So whether is it effective or not, or harmful or not, then I would not be so much worried, because all the actual effect sizes are on the same side, they are all significant and all have the same direction of effect. However, if our original research question in our systematic review is about the level of efficacy, then I am worried, because then you would see that we have 2 subgroups of effects. There are groups which are as big as here, and there are groups with much larger effect sizes. So what you always need to do when you try to interpret the certainty of the evidence is, again, go back to the body of evidence, but reflect to your original research question. So I hope it's clear. If it's about efficacy, I'm not so much worried. But if the question is about level of efficacy, then I am worried here. Okay, the next factor or domain to assess certainty of the evidence is imprecision. So for imprecision, we actually need the meta-analysis, and we need to look or to assess how precise the actual overall effect estimate is. So generally, results are imprecise when they are based on few animals or few events, for example, leading to very wide confidence intervals around the summary effect estimates. And we think that often results are precise when there are the optimal information size is met, which means that there are actually enough animals, enough studies, and the confidence interval excludes no effect. And we are also thinking that the results are precise when the confidence intervals overlaps with no effect, but the confidence interval is very narrow. Now, I will try to show you this in the next example. So this figure may be helpful to actually understand what I was saying. So when we think about imprecision and you see this graph, you need to consider this as diamonds. So they are not individual studies, but this is the summary effect size of your meta-analysis. And these first three diamonds, in a way, so overall effect sizes, they show precise effects, and I would not downgrade. However, the three here below in reds, they are all imprecise, and I'm going to explain you why. Now, this is an example from the clinical field, and here you see the zero effect line. This is the zero effect line. And in this case, they also had a clinical relevancy effect, which they are taken into account. In animal studies, we're not sure yet how to deal with that, but for practical reasons, I will now explain how to work with it in a clinical setting. So what you could see here is in the first diamond or overall summary of the effect, we see a diamond which excludes, the confidence interval excludes zero, which means that this is a significant effect. And it does not include, the confidence interval does not include the clinical relevancy line. So we are very certain that there is a non-relevant significant effect. The second study is also very precise, but precise about no effect. And that should be clear by now that there is the zero is included in the confidence interval, so it's not significant. And the clinical relevancy lines are not included. So it's again, it's in this case not significant and not clinically relevant. However, this third diamond may surprise you because here you actually now see a overall summary of the effect with a very wide confidence interval, and I still would not downgrade my evidence or the certainty in my evidence. And this is because zero is clearly not included, but also in either the left or the right side of my confidence interval, always the actual effect is clinically relevant. So although there is a wide confidence interval, the effect is in 95% of the cases at least clinically irrelevant. So in other words, if you now look at the the diamonds in red, you now understand that this first one includes the no effect line and also the clinically relevant line. So we're not sure about the actual effect. It could be clinically relevant, it could be not effective, and it's definitely not significant in this case. The second one is a much smaller confidence interval, but includes the clinical relevancy line. So again, because of that, we're not sure about whether or not this effect is relevant. And for the third one, it's a wide confidence interval. And again, we're not sure, although statistically significant, whether or not there is a clinical relevant effect. So again, here, a lot of words to explain to you that it depends on the body of evidence and the type of research question, whether or not you would downgrade this, the certainty in the evidence. So sometimes the clinical relevancy lines are much closer to zero, and a very small effect may be already important. Okay, the last downgrading factor is publication bias, and I already mentioned it briefly. This is about studies that are not published or unpublished parts of studies that may cause an overestimation of our actual effect size in a meta-analysis. So, as mentioned, the reliability of the results can be diminished by unpublished studies. And in order to assess what the actual risk is, we can create and interpret a funnel plot and impute the missing studies. And we generally already do that in our systematic review. However, then we still need to do the interpretation part. So how much are we actually worried when there is risk, there is publication bias? So it is not necessarily the case that when you have publication bias that you would also downgrade your evidence. It actually depends on the effect size. So it needs to be again relevant for your actual effect size. So below here you see a systematic review for which they recreated a funnel plot. And here you see the studies with low precision on the bottom of this plot, and the studies with high precision, they are more close to each other. So you have an upside down funnel. And the actual effect of the meta-analysis was a 20% increase of a specific intervention. And now we assessed with a trim and fail analysis whether or not there were missing studies. And all these red dots, they suggest the potentially missing studies. And if we take that into account in our meta-analysis, we can see what the actual effect would be if we would have found these studies. And in this case, the effect now reduces to 14%. So this is where grading or downgrading starts. So what we now need to assess is how serious do we think that this reduction in 6% is for the interpretation of our study results. So sometimes it can make the difference between a relevant and a clinical relevant and a not relevant result. It can make the difference between significant and not significant, and then it's quite obvious, of course. But the challenging part is try to assess how serious this affects the certainty of the evidence, of the body of evidence. And I was lying before because not publication bias was the last one. Indirectness is the last domain for downgrading. So when we're talking about indirectness, we're talking about a population studied, if that's actually also the study population which we're interested. So when we're talking about direct evidence, we're talking about research that directly compares the intervention in which we are interested, in the population in which we are interested, and measures actually the outcomes important to patients. Now, in the context of preclinical animal studies, we have two layers when we try to assess whether or not there's indirectness in our systematic review. The first layer is about the actual preclinical research question we had in our review and the evidence that we summarize. So that's quite clear. That's the evidence that we're working with. And layer 2 is about the evidence we were working with and the translatability to the actual clinical situation. So again, here we're talking about the body of evidence, and we need to determine if the presence of indirectness influences the reliability of our conclusions. So a few examples about indirectness to make it a little bit more clear. So for example, if we talk about the first layer, so the preclinical research question and the observed evidence, you could think, for example, about indirectness when you have a preclinical research question which is focused on the effects of drug X on vascular calcification, but the studies assessed in our systematic review, they focused on vascular events, which is a consequence of calcification. So then we have an indirect outcome in our systematic review. Another example could be in this first layer that we have, again, a preclinical question focused on outcome X in animals with stroke and comorbidities, but in the identified evidence, mainly animals without comorbidities were used. And you will often see this in the preclinical studies that are conducted. When we're talking about layer 2 of indirectness, we're talking about the actual translation from animals to humans. So for example, in the preclinical studies, histological damage was determined, and it was a surrogate for functional loss in clinical studies. And another example which we had in a systematic review of our own was that the timing of the intervention in animal studies was actually before inducing the disease, and in patients it's often after the disease. And this was an example, for example, about the effect of probiotics in acute pancreatitis. And in all those animal models, they first induced the disease. No, they first provided the medication before inducing the disease. And in patients, you first arrive at the hospital with your acute pancreatitis, and after that, you receive the drug. And these type of indirectness, all the different types of indirectness that may be relevant, should be described in your discussion and should be clear and assessed. And actually, again, we need to assess how much this downgrades the certainty in the evidence we have regarding the diamond of our systematic review. There are also a lot of upgrading factors, and I will only briefly discuss them because I see that I'm talking already for half an hour, that I need to speed up a little. There are also some upgrading factors, as I mentioned. You may want to upgrade, as I mentioned before, when the meta-analysis shows a clear dose-response relationship, or when there is consistency between all these species. So here, for example, you see an effect in rabbits, in mice, in rats, in pigs, and they all show the same direction of effects. So there is still a lot of heterogeneity between those species, but the direction of the effects is similar. And this will provide us an additional confidence in whether the effect is the same in human species. Now, when you finally assess all these factors I just mentioned, you can now create an evidence profile, an overview of the certainty of the evidence. So here you can see, for example, an evidence profile of randomized clinical trials. Here you can see the question, and it borrowed again from the clinical field, about the effects of antibiotics for children with acute otitis media. And you can see that the quality of all factors is graded per outcome. So you see here risks of bias, which they call limitations, and they didn't downgrade. Inconsistency, they didn't downgrade. Indirectness, they didn't downgrade, etc. And then in the end, they reach an overall quality rating. And in this case, for this outcome about pain, they decided that it's a high certainty in the evidence regarding all those factors that may have downgraded, potentially could have downgraded the evidence. And what is important is that you do this rating, as mentioned before, for all outcomes separately. Now we know actually how GRADE works, and I hope that it's clear that we need such a structured way of interpreting the results of preclinical systematic reviews as well, because it's very challenging to make sure that all the factors that may play a role are actually taken into account. And in 2016, Miranda Langendam and myself, we started to think about how we could create something like this for preclinical animal studies. And in 2018, we published a preliminary GRADE-like framework. And this framework that should help us when trying to decide whether there is sufficient therapeutic potential of a medical intervention to proceed to clinical trials. So that was the main purpose, or whether there is still need to conduct new animal experiments based on the existing evidence. Now, this system in this paper is still in its infancy. We know which domains you need to take into account, and all those domains I just described, but we still do not actually know how much to downgrade or upgrade for each specific element. So I will say that again in a few minutes, but for now it's really important to just make sure that you know which domains that need to be taken into account, and describe these one by one in the limitation sections of your discussion per outcome. So the actual GRADE approach for assessing certainty evidence of preclinical intervention studies was developed in the context of therapeutic interventions. So what we mean is that this framework for experimental animal studies are studies in which the investigator controls the intervention. So that's essential because, as I mentioned in one of the earlier slides, is that all these GRADE frameworks, they are really focused or specific for different research fields. And this framework was for these controlled studies to test efficacy and safety of medical interventions. So that's important to know. And here you can see the actual approach. So step 1 is framing and the rationale of the clinical question. So not the preclinical question, but in this framework, we first need to know what our actual clinical target was. And therefore, you need to use the basic PICO methodology. Subsequently, in step 2, you can start with scoping the evidence. So in the second step, we try to assess whether or not there is available clinical evidence. And only when there is no evidence or very limited evidence from human studies, the GRADE approach for animal studies can be used to answer the question using evidence from animal studies. So if this has been done, step 1 and 2, we actually see the need for this preclinical systematic review. We formulate the preclinical PICO. So this it is important to remember the clinical question is leading, and the preclinical PICO should be derived from the clinical PICO. So a separate preclinical PICO is needed to define the eligibility criteria of our systematic review of animal studies, and it also serves as the first outline of aspects of indirectness, because in step 3 we have step 1 the clinical question and step 3 the preclinical question. In step 4 then we can finally collect the preclinical evidence and summarize the effect estimates by outcome. So in a way we conduct our systematic review, and the eligibility criteria, as mentioned, are borrowed from the preclinical PICO, and then all evidence ideally would be summarized in an evidence profile, as I just showed you. In step 5, we assess the quality of the animal research per outcome, and in this approach, controlled animal studies start as a high quality of evidence. So if you remember one of the previous slides, observational studies often start with a low ranking and controlled studies start with a high ranking. In rating the certainty of the evidence, we propose to assess first by outcome the grade downgrading factors, and then we start with risk of bias, imprecision, inconsistency, publication bias. And then secondly, when this all is done, we assess the 2 layers of indirectness. And finally, in step 5C, we consider whether upgrading may play a role. In step 6, that's Dutch. In step 6, we rate the certainty in the evidence. So in order to finally rate the certainty of the evidence in either high, moderate, low, or very low certainty of the evidence, all these grade factors need to be considered simultaneously. And the biggest challenge for now is that it's not clear, as I mentioned before, how to weigh the various factors. For example, how much weight should all these factors individually have in the overall rating? So, as mentioned, this framework is just in its infancy, and there's a lot of steps to be taken. And the 2 most urgent topics on which we are currently working in an international setting relate to inconsistency and indirectness. So regarding inconsistency, the main issues are that that part of the heterogeneity that we have in a systematic review of animal studies is intentionally induced, because animal studies are explorative by nature. So by nature of these animal studies, we generally have more heterogeneity to start with than in clinical studies. And we want to know how to deal with that. Should this unintentional heterogeneity be excluded, and how do we do that? So that's one of the issues, and the other issue is that we think that there are different levels of inconsistency present, not only within species, so you can have a lot of heterogeneity between those studies, And this can be within one subgroup of species, and it can be across species. And how to integrate these two levels of inconsistency in a framework. So this is where we are actually working with in the GREAT working group. And we have a specific project group on animal studies. And yeah, as mentioned, in a international collaboration, these are the main issues. The other item is regarding indirectness, which is probably not surprising for you. This is very challenging because we don't know how to determine which aspects of indirectness need to be taken into account, and we don't know how to weigh them. So, for example, in blue, in this slide, you see one of the questions I may have. So, for example, you have a study or a subgroup of studies that use a lower animal model and representing the same metastasis-associated protein as humans. So, for example, transgenic mouse. And does this model better reflect clinical pathophysiology than a higher model, much closer to humans, but expressing species-specific metastasis-associated protein? So, for example, a pig model. Now, we have all these different aspects of indirectness playing a role in multiple models, and we need to be aware that there is generally not one model that reflects all aspects of the clinical disease, and we need guidance on how to deal with that and how to take that into account. But for now, I presented domains, I presented the challenges, but what could you do? Now, what you could do for now is make sure that when you conduct your systematic review and you try to interpret your results, that the factors below will be described or descriptively taken into account in a limitation section of your systematic review, and try to do that per outcome. So when you try to interpret your results, take into account risk of bias, heterogeneity, imprecision, indirectness, and maybe overestimation due to publication bias, and consistency between species. As a last thing, in case you're interested to work on this topic, join the GREAT for Animals project group. If you want, you can contact me. And as a last take-home message, I hope that at least from all this information you take home, that GREAT is a structured way to interpret the results of your systematic review and reliably evaluate the potential benefits and harms of a treatment and try to balance that. GREAT needs to and can be applied to preclinical animal studies in the context of therapeutic interventions, and there are a lot of methodological challenges to be resolved. I think that there are other things that are important, but those are the main important things. And although we don't know if we downgrade for 2 levels or for 1 level, you can always discuss at least these factors or take them into account in a structured discussion section of your own systematic review. So thank you for your attention so far. Yeah, are there any questions?